Good afternoon, everyone. One of the privileges of being NIH director is the chance to come here on Wednesday afternoon and to introduce the Wednesday afternoon speakers, and we are fortunate to have, week after week, a remarkable number of national, international leaders in their fields of scientific research, and I'm glad to see all of you here, and also want to say welcome to all of those who are watching on the web, who are probably even more numerous, because that's the way people seem to do things these days. It's a particular privilege, though, to be able to introduce a speaker when it's somebody that I have known well, in fact, worked with closely over the space of something like 20 or 25 years, because today's speaker, Elaine Mardis, has been a central figure in the revolution in genomics over that timetable in her critical roles at the Genome Center at Washington University in St. Louis. I suspect she has some PTSD from some components of that experience as well. I didn't ask you, Elaine, whether when it's 11 o'clock on Friday morning, whether you have this sudden tightening in your chest because it's time for the weekly conference call of the genome sequencing centers to find out whether we're on track or not to actually finish the Human Genome Project. I still have that experience on Fridays at 11. And having been a major part of that effort at WashU as one of the 5 centers that did almost all of the work, I suspect that leaves a mark. But happily, it leaves a mark now that one can point to with a great deal of excitement in terms of where we have gotten to with the field of genomics. And Elaine is a terrific person to talk to you about that today, particularly as it relates to cancer. Very briefly, she got her Ph.D. at the University of Oklahoma with Bruce Rowe. She worked for 4 years in the private sector and then went to WashU, where she has been since 1993, and is currently the Robert E. and Louise F. Dunn Distinguished Professor of Medicine. She's professor in the Department of Genetics and Molecular Microbiology and co-director of the Genome Institute. She's received a number of awards and honors, including the Scripps Translational Research Award for her work on cancer genomics. And she will receive the Morton K. Schwartz Award from the American Association of Clinical Chemistry for significant contributions in cancer research diagnostics this year. Elaine has been not only a contributor to remarkably large-scale production efforts, she has always been an innovator, an inventor, somebody who is looking at the technology as, in many cases, the most important part of our portfolio, recognizing that many of the things we wanted to do and want to do now were unaffordable unless the technology got better. And the ways in which she has contributed to the fact that a human genome can now be sequenced for roughly $1,000 are very numerous and very significant. So it's a great personal pleasure to have her here, and I would like to ask you now, please welcome Dr. Elaine Mardis.
So good afternoon, everyone, and thanks for coming. It's really a pleasure to be here, and thanks especially to Francis for the kind invitation to present here today. I guess in broad brush, what I'd really like to present today is a sense of excitement about where we're going with large-scale cancer genomics discovery, and to give you a feel for some aspects, certainly not all because there's not enough time, about the directions in which we're beginning to take the broad brush strokes that we've learned about cancer genomics, the drivers, the important genes, and other factors from large-scale discovery, and transition now how we're beginning to approach cancer patients in the clinic. So that'll be sort of the theme for today. And I'll paint that in broad brush strokes across both the blood-based cancers, focusing on acute myeloid leukemia, and then the solid tissue malignancies as well, looking at melanoma and ultimately brain cancer. So I'll do that hopefully within the allotted time, and we'll have time for some good questions after that. So I want to first focus on the efforts that we've had going on for quite some time now in acute myeloid leukemia, as I said, and then transition more to new ways that we're looking at the genome in the context of novel immunotherapeutic interventions for patients. So studying the natural history of AML has been a focus of our efforts for quite some time now. As I mentioned, we first started this work in 2007, just coming out of the Human Genome Project, where we literally had the ability to identify most, if not all, of the genes in the genome, and to go after that list of genes that we thought would be important in the a disease known as acute myeloid leukemia. Part of the reason for doing this initial look at the leukemia genome was because we really wanted to better understand how to stratify patients. You can see from this Kaplan-Meier curve put together in 2002 and largely unchanged today, although I'll tell you more about how we hope to change this, that most patients in the disease have so-called intermediate risk. That means that based on any of the known markers such as chromosomal translocations, chromosomal complexity, or other factors such as age, etc., we really don't know how these patients will do in the disease. And overall, as you can see from the curves here, most patients do die of their disease. Very few, about 17%, have what are known as favorable risk outcomes, due largely to chromosomal translocations such as the T1517 in acute promyelocytic leukemia that predicts a good outcome based on current therapeutic interventions. So at the time, 2007, we knew very little about key initiating mutations for AML patients. These intermediate risk patients overwhelmingly had normal chromosomes, so if you looked at them under the microscope, there were not widespread aneuploidies, no indicators there, and very few deleterious somatic mutations. And ultimately, we really just were looking for prognostic markers, ways to stratify patients into better risk or poorer risk, so that we could then determine how aggressively to treat them or not. And in this effort, circa 2007, we essentially, as I said, made up a list of candidate genes, designed primers to amplify those out of AML genomes using PCR and compare to normal, and begin to look at mutations that were potentially causative in the disease. We only got so far with that before next-gen sequencing came along. And this, of course, was not immediately applicable to whole genome sequencing, but there weren't, at this point in time, good partitioning methods, such as the exome or panel hybrid captures that I'll talk about later. Instead, we really only had one option, which was to go after the whole genome. This was a fairly expensive proposition at the time, so sequencing a whole human genome on these early generation Solexa sequencers was a pretty sort of dramatic task. First of all, we didn't know what the right coverage was. So how much do we need to oversample the genome to get coverage across all of the human chromosomes to the depth where we could identify mutations? Secondly, we didn't know what bioinformatics to use, so how best to compare tumor to normal once we actually had the data in hand, and so on. And so we took a fairly simple case here. This is an idealized case, a patient with highly diploid chromosomes, almost 100% tumor content in the bone marrow aspirate that's shown here on the H&E that was taken at diagnosis from this patient. So we didn't really have to worry about contaminating normal signatures in the genome sequence. And we really, because this was such an adventurous project, went out to obtain philanthropic funding for this single patient genome, which we were fortunately able to do from a local donor, Al Seitman, who also funds our NCI Comprehensive Cancer Center. And this is really the $1 million experiment. So the data generation alone for this patient cost on the order of about $1 million using current technologies at the time, Solexa sequencers that would only generate 1 billion base pairs per run. We calculated an average coverage that was needed of about 30-fold. That turned out to actually be about the right number. And from that initial study, we identified 10 genes with mutations, 2 of which had been previously described in AML. So we weren't sure if 10 was the right number either, but now having sequenced through over 200 AMLs, as I'll talk about later, we now know that on average, AML patients across the spectrum from favorable to intermediate risk average 12 mutations in their genes. So we weren't too far off of the average at this point in time. Progressing forward then, using this whole genome sequencing paradigm, we developed a number of nuances that I'll tell you about just in the context of one thing, which was verifying that our mutation calls were actually correct. So keep in mind that the bioinformatics still of whole genome sequencing are a bit in development, although at that point in time they were entirely nascent. What we knew that we could do, especially once we got to the point of being able to do paired-end sequencing, which was not done on that original AML genome, is that we could sequence from the tumor about 50-fold, so oversampling was better for coverage in identifying mutations, sequence from the normal about 30-fold, and work through a series of algorithmic approaches to the data that are shown here in these different boxes once we had aligned all of these read pairs to the human reference genome, tumor to normal, identified changes, and then compare one to the other to detect what's truly somatic. Beyond that, then we really developed some methods to double check ourselves to make sure that we got it right the first time around. And that really went to designing custom capture probes that represented each variant site that we had predicted in the genome, not just single nucleotide and indels, but also, complex structural variants where we would literally tile probes through the variant region, hoping to capture that back out, resequence those, and double check those on alignment to the genome just to make sure tumor to normal that we had the right variants that were identified. And iteratively, as we did this over and over again, we were able to now build filters into our calling pipelines to actually tease out known sources of false positivity. So we became much more precise in the process of doing this. The other thing we finally realized as we were going through this exercise was that these data were valuable for another reason, and that is to evaluate the heterogeneity, the cell-to-cell based differences in cancer masses that occur by virtue of comparing deep sequencing data at all of these variant sites that were captured by hybridization. So I'll show you how this worked and then show you some examples from a single patient study. This is what we refer to as DDS, or deep digital sequencing. What you do is what I just described a minute ago, design custom capture probes for all of these sites in the genome that you think contain variants. You hybridize those to the tumor or normal genome that you've already sequenced through in the process of generating the data the first time around. And these hybridized fragments can be pulled down by virtue of the fact that the custom capture probes have biotins on them and can be captured out of solution using streptavidin-coated magnetic beads. And once these are isolated away from the rest of the genome fragments, post-hybridization, these are readily eluted off and then amplified in sequence. So you can essentially just recreate very high-depth, isolated regions of the genome that you're interested in evaluating a second time for variants. So because of the high depth that is generated on these, and because of the nature of next-gen sequencing being very digital, each read on a sequencer essentially represents a single fragment from the library that you've sequenced, we can now go about estimating heterogeneity for any population of sample, any population of tumor cells that have contributed DNA into a sample. So the way this works is that you generate this high-depth coverage that I've already shown, and by virtue of aligning back to the genome, you now have a digital readout of each of the two alleles that are present in the population of fragments. So each mutation will show up typically as a mixture of wild type and mutated reads, because most regions that we identify are in fact heterozygous mutations, although not all. And then you sort of begin to look at counting the digital representation of each variant, where by inference, the older mutations that are present in every cell, basically the founder clone that went on to form that tumor, are present at very high varying allele frequency. So for heterozygous mutations, this approximates 50% of the reads being represented carrying the variant. Whereas any newer mutation in the tumor cell population will be present, but at a lower variant allele fraction. And so fewer reads in that population of captured fragments will represent the variant. We can then use some statistical Bayesian methods to essentially model tumor heterogeneity by grouping together variants that share the same approximate variant allele fraction, and then build a model of the clonality or heterogeneity of that tumor. And I would argue that, as I'll show, just looking at a single tumor in isolation in terms of heterogeneity, kind of interesting. Actually looking at the changes in heterogeneity or clonality over a natural history of that tumor, much more interesting. So what is the impact of therapy, for example, on the changes between primary disease and treated disease, for example, or the primary versus the metastatic setting. So I'll show you some examples of that from our work with AML. I'll show it in a couple of ways. Graphically, we'll sort of look at comparison of the variant allele fractions between genomes, where in this particular toy example, I'm looking at tumor 1, that could be the primary presentation, and tumor 2 could be the relapse, for example. And graphically, this is the founder clone that I've just described. So those set of mutations that are present from the beginning of the tumor's occurrence towards progression and are shared by every cell in the tumor. Anything that's new, for example, in the relapse or metastatic setting would group closer to this axis, and anything that gets left behind by virtue of the therapeutic intervention would group closer to the tumor 1 unique axis. So this is the toy example, and we'll see how that plays out in real samples in a minute. The other way to illustrate this for those of you who are graphically oriented, and many people are, because this is a figure from a Nature paper that has been used not by just me and my colleagues, but many people over time and replicated in other papers by other groups to really illustrate the progression of a tumor throughout evolution, which is really a truly evolutionary process. So in this particular example, which is from this study in Nature 2012 of 8 primary tumor relapse samples in AML, what we're able to identify for this patient, again, the first patient that we ever sequenced in that Nature paper from 2008, by studying her relapse disease and at very high coverage her tumor in the primary treatment-naive setting, we identified that this patient expanded one of her hematopoietic stem cells by virtue of a suite of founder clone mutations that are listed here, DNMT3A, NPM1, FLT3, and PTPRT. And these flourished in the bone marrow to create 4 different subclones that are present in different proportions. The highest one at 53%, it has its own unique genotype on top of the founder clone, and these other 3 minor subclones that also have their own unique genotype on top of the founder clone. You can now see the incredible evolutionary pressure that's put on this genome by virtue of the chemotherapeutic intervention that's used to try and get this patient into remission. And only by sequencing the relapse clone do we identify the fact that this minor subclone, present at about 5% in the primary disease, acquires additional mutations, a translocation that fuses 2 genes together, and now flourishes as the sole population in this patient's relapse sample. So it's related to the primary, but it's a minor subclone, and we saw this in multiple patients in this study, where we were still at that point fairly nascent in our ability to sequence deeply, and so many of these patients appeared to be monoclonal, but actually probably had very minor subclones that we just could not detect. So let's dig deep into one of these patients that was originally presented into this study, and I'll show you sort of the natural history of the disease, focusing on this one patient. This is a patient AML 31. You can see a fairly typical presentation in AML, the treatment regimen, achieved clinical remission from this regimen, but relapse after 16 months' time. And of course, the beauty of studying AML is that the clinical protocols, the standard of care, permit bone marrow aspirates to be taken at different time points in the disease. And if you have the banking protocol, you can save these over time and go back, as I'll show you, to investigate the natural history of each patient's disease. So in this initial Nature paper, we just did fairly standard low coverage genome sequencing at the whole genome scale was still pretty expensive in 2012. So we studied the normal, tumor, and the relapse disease, and then went for high coverage capture validation that I just described for you. And we were able to sort of put together this particular diagram of this patient's cancer evolution. So the primary disease here with 3 subclones, a founder clone and 2 minor subclones, and then the recurrent disease with just a single relapse genotype present. We then sort of put this particular patient's cancer genome on steroids, if you will, in this paper that just came out in 2015 in Cell Systems. I think this was actually the first issue of this journal. And this is the sort of graphical abstract. And you can see here that what we've done now is we've gone well beyond the sort of standard 50-30 fold coverage that I described for you earlier to address a number of questions, which I'll walk you through fairly quickly because this is already published. So you can now see that we really kind of went a little bit crazy on this in terms of high depth coverage, whole genome, exome, this deep capture validation that I've told you about already, targeted sequencing using 260 recurrently mutated genes in AML, more on that later, digital droplet PCR, lots of RNA-seq data, et cetera. What did we learn from this study? Well, first of all, we learned a lot about different short read aligners and different variant colors. And we learned about what we could find from orthogonal validation data, including digital droplet PCR compared here to sequencing, variant allele fraction, pretty good concordance. Ion torrent data versus Illumina-based capture data, not so great concordance. And interestingly, and I'll get back to this at the end of the talk, the correlation between DNA variant allele fraction and RNA variant allele fraction, which interestingly, even for a case like AML's, where there are very few mutations that are actually identified, some of these mutations clearly are not expressed at the level of RNA. More on that later when I get to melanoma. Coverage depth definitely matters. Can we afford to sequence every tumor to 300-fold coverage? No, we cannot. And even in the $1,000 genome era, we really can't afford to do that. But I think you can readily appreciate moving from panel A to panel F here, the impact that additional coverage has on really tightening our understanding of clonality in the disease, where different clonal populations become much more evident as we pile on coverage for this particular tumor sample. And here's sort of the final delineation through our Bayesian mixture modeling algorithm called Cyclone, where interestingly our platinum call set, so highly validated mutations through all of these different exercises that I just showed you, turns out to be on the order of about 1,300 point mutations, single nucleotide variants, genome-wide, compared to only 119 that were originally identified from the 30-fold coverage of the genome. And again, you can see beautiful examples of known driver mutations, such as FLT3 IDH1, clearly dropping out in subclones that are defeated by the induction chemotherapy in the primary disease, and new ones that are occurring, interestingly now, in this subclone that's unique to the relapse, an IDH2 mutation in the hotspot location. So lots of interesting vignettes here, and of course, number one, as I'll just point out to be obvious, is the founder clone, which persists in both presentations of the disease. Now our tropical fish plot looks a lot more complicated and representative of the heterogeneity here at day 0 when the patient is diagnosed, and at the recurrent sample here in 500 days down the line. And you can see that we've now identified key new mutations in all of these different subclones. Back to the banked samples that we had in the freezer at different time points from the initial diagnosis all the way to this relapse diagnosis that I've already told you about. And through high-depth sequencing using digital droplet PCR, looking just at specific mutations, you can sort of see the ebb and flow, but never the complete diminution of a mutational signature in this patient. In other words, high-depth sequencing of this type can actually give us a clue about whether patients are indeed in remission or just have some occult disease that can't be detected by commonly used pathology methods. The other interesting vignette in this patient's genome reflects something that's now sort of widely appreciated, and that is that patients, even without leukemia, can develop hematopoietic stem cell expansion. So they acquire just a single mutation that's a growth advantage, if you will, for that hematopoietic stem cell. And even though they're still completely healthy, every one of their developed blood cells reflects the genotype of this single hematopoietic stem cell example. In this particular patient, this was a p53 mutation that was acquired, and you can see better is this sort of bright blue subclone. This is a hematopoietic stem cell expansion that occurs while the patient is in remission, or apparently so. It's not cancer, because it's completely unrelated to the genome of the cancer. So you can see these are originating from two completely different HSCs over here. And this expansion phenomenon essentially goes away when the patient recurs with the relapse disease at day 505. So just to finish up with this particular vignette, we've now completed a number of samples in the third iteration of their tumor. So once these patients relapse in the disease for the very first time, they go through induction chemotherapy to remission and receive a stem cell transplant, a so-called allogeneic stem cell transplant from a related donor if one exists. Many of these patients will go on to develop AML a third time. And so we've now completed a study, not yet published, of allotransplantation disease after the first relapse to really address the question of what is the genomic makeup of the allogeneic or the second relapse disease, rather. And so this patient is no stranger to this approach. As you can see, we've got the second relapse banked. We did exome sequencing in our clinical pipeline, which I'll tell you about in just a moment. We identified 41 mutations above and beyond the 11 that were previously known from the first relapse. These are in genes in particular, since this is exome sequencing. And the patient is now deceased, unfortunately, is the outcome for most AML patients in the disease. And now you can see sort of the full picture, if you will, slightly different color scheme, sorry. But this is now the TP53-driven clonal expansion that I was telling you about just a minute ago that diminishes at this first relapse. And you can also see that the second relapse disease includes this green founder clone present already here, but at very low levels, and a minor subclone or two, answering the question, as we did with every one of these 12 samples in the study, of the fact that the second relapse disease ultimately is related to the original disease by virtue of the mutational spectrum. So even though you've been implanted with another person's marrow, essentially, the disease you get is the disease that you started out with some changes as obviously reflected here. So where has this brought us to? I think, first of all, we had to go through a large-scale discovery phase, which I'll just tell you about briefly. This was through TCGA. All 200 of these samples that were studied in this project were from our tissue bank, at our bone marrow bank, rather, at WashU. And this was a New England Journal paper just a few years ago, looking at all the multi-omic platforms that one did through TCGA. What did we find? A whole lot of stuff. So I love and hate to put up figures like this because they're so complicated. But the bottom line here is that AML, like many cancers in the solid tissue setting, finds myriad and numerous ways to invent itself by altering the genome. And this is just genomic alteration. So it says nothing about changes in methylation, RNA editing, or anything else. What were the surprises here? Well, just a couple. First of all, spliceosome-based mutations were not suspected to be drivers in cancer. This has now sort of opened up a whole new field of effort, if you will, in looking at what the impact of these mutations on proteins that are part of the spliceosome has. And you probably can't see this, I can't, but cohesin mutations as well are of great interest, where cohesins are essentially involved in partitioning chromosomes at cell division and turn out to be highly mutated in this disease as well as a couple of others. The bottom line is that when you do these large-scale studies, you then progress quickly from this global view of what's going on to some sort of statistical analysis of significance. In other words, which are the genes that are mutated on average more than you would expect? And so this was sort of the list for AML, being a low mutation load tumor. The list is not large, 23. And even though 200 samples sounds like a lot, it's not really a lot. It was across all of the subtypes for the most part of AML. So we feel like we still have more to learn. But rather than spending time sort of doing additional discovery genomics, what we did with this list, as well as a list of additional genes that were not statistically significantly mutated, but recurrently mutated, or RMGs, is we generated a list of 259 genes, had a probe set of capture probes synthesized to correspond to all of these genes, and decided to go after clinical aspects of AML, as I'll tell you in a moment, using the combination of these RMGs, recurrently mutated genes that include this list up here on the graph, and exome sequencing to sort of have our cake and eat it too. We wanted to do additional discovery in all the genes, but by spiking in additional probes from these RMG genes, we actually up the ante in terms of coverage. More probes equals more coverage. It's stoichiometry when it all comes down to it. And this would give us additional coverage so that we could do heterogeneity change analysis like what I just showed you from the discovery of the AML natural history. So now we're moving this type of assay into the clinic, and of course, you can't just do genomics. You have to pile it in on top of the clinical reality for patients so that you can see whether it makes a difference or not. So here's how we're planning to do that. These are kind of the spectrum of possibilities when you're diagnosed with AML, if you will, in a simplistic view. You either have completely primary refractory disease, that means it never responds to chemotherapy and you don't go into remission, or these vexing patients with early refractory disease that appear to have responded well to their induction chemotherapy but actually relapse within about 6 months' time or less. There's an intermediate group, fairly rare, and then even rarer, patients with long first remission or prolonged first remission. So this remains a highly deadly disease for which treatment paradigms really haven't changed in the last 25 or 30 years. We do know that there are some predictors, and we want to add to those or see whether we can add to those using a specific genomic analysis, which I'll tell you about. The challenges here are that we have imperfect prediction of relapse potential. Now, even 7 or 8 years after the initial studies that we started doing, and others, of course, are playing in this field as well. No indicators of patients who will be primary refractory, unfortunately, and we still don't well stratify patients with intermediate risk. The other thing to point out with respect to the age component is that most AML patients, like most cancer patients, are old. And so this is a disease of old age where, quite frankly, the severity of induction chemotherapy, if you're unfortunate enough to be primary refractory, your ability to survive a second, even more aggressive round of chemotherapy trying to get you into remission is sometimes very, very slim. And so one of the things that we would like to try and do with this genomic assay that I'll tell you about is potentially identify other therapeutic modalities that might be alternatives for these patients as a clinical trial or in combination with another round of chemotherapy to help get them into remission. One of the ways of doing that, I just have to put in a shameless plug for this database that we're building along the lines of connecting genes with mutations to causality in cancer, is our CIVIC database. And we've been putting this together for a couple of years now. We're just getting ready to publish this, but it's available already at this URL that you can see here. So please feel free to take a look. This is really just an effort to make a publicly available database for people to crowdsource information into. So we'll be opening up the crowdsourcing component soon. Already we're annotating clinical variants in here with evidence from the peer-reviewed literature, both for preclinical evidence as well as for treatment-based evidence from clinical trials. And it's really just an effort to coalesce a lot of information about variants, genes, treatments, prognosis and diagnosis as well. And so please feel free to take a look. But this is the type of resource that we will build additionally coming out of the clinical trial that I'll tell you about now. So the clinical workflow that we came up with for AML patients, as you can imagine, is a little bit different than what you might think about or have heard about for solid tissue malignancies, where typically a biopsy is taken, a panel or an exome is done, and we evaluate for a single point in time a single or multiple therapeutics that might be applicable to that patient in the fight against their disease. In AML, the paradigm is different, as I've already explained. We have patients who are being diagnosed at day 0. There's no waiting for these patients. They go as quickly as possible into clinic. This is all done in the hospital setting because of potential complications, and they begin their induction chemotherapy pretty quickly after they're diagnosed. At the end of day 30, they're done with their induction chemotherapy round, and they should be reestablishing their marrow. This marrow is typically harvested through the hip and evaluated under the microscope for evidence of minimal residual disease. So these are the two samples that we'll focus on in our clinical study, day 0, day 30. The comparator here is a skin normal, or if that's not available, we've actually developed a mechanism for tongue brushing, which gives us less bleed through from the capillary beds in the tongue, where the key is to not get white blood cells that are circulating often at high numbers from the patient's blood supply. And I won't talk about it, but we'll also be doing RNAseq from the day 0 and day 30. The reason for doing this is to identify occult fusion transcripts that may be there, but for which a chromosomal alteration may not be identifiable from cytogenetics. So using the DNA from these 3 samples, we'll apply our exome plus recurrently mutated gene spike-in hybrid capture approach, and then we'll do high sensitivity variant detection, where day 0 gets compared to the skin normal, and day 30 gets compared to the skin normal, and then we compare what's changed, if anything, about the variant allele fraction between day 0 and day 30. We compare the mutations and we compare the variant allele fractions. So before you sort of embark on this type of a study, as you can imagine, and given the value of these bank samples that I told you about earlier, the smartest thing to do, I would argue, is to apply that assay, shown here, to samples for which you already know the outcome for the patients and for which you have the day 0 and day 30 marrows already banked. And so we did exactly that. This is, again, just recently published at the end of last year in the Journal of the American Medical Association. And I've just showed 2 patients here for the purpose of examples. So this is the patient that you want to be, I guess you could say, if you're unlucky enough to be diagnosed with the AML. You can see the founder clone mutations here, including a FLT3 point mutation, NPM1. These drop to essentially undetectable levels at the day 39 marrow for this patient when their bone marrow aspirate was taken, and maintain this undetectable level out to the last point in time that we had a marrow available for this patient. Are we infinitely sensitive to identify mutations? No, we are not. We're probably sensitive down to about the 1% level, using about 500-fold coverage from this RMG spike-in. So just want to be clear about that. But this patient is more sort of typical, if you will, of most AML patients. So lots of mutations can be identified here in known drivers. In the primary diagnostic setting, after the induction chemotherapy, some of these have dropped to baseline, but many of these are still at the detectable level in this bone marrow aspirate. And indeed, not surprisingly, this patient presents with recurrent disease in a fairly short time window with the exact same mutational loads to a large extent that they had to begin with by testing in the primary setting. So now when you take these 2 patients and add in lots more, what do you get? Well, it looks sort of like this, where, again, this is event-free survival longer than 12 months. So these are our long first remission patients, and these patients are the either primary refractory patients, where there's really not much change at all from day 0 to day 30 and ultimately out here, or these patients will come back very quickly in a short time frame with recurrent disease. So from this retrospective study, we've now got a CLIA assay developed and evaluated and has the stamp of approval of CAP. And we're currently enrolling patients on this study in our clinics. The idea is to bring in every AML patient, regardless of their FAB classification, just to see what we can find. So this is sort of a learning exercise, but done in the CLIA setting. And I've already described the assay for you. What I didn't tell you is we'll put this up against a very hard target, which is a commercial entity that will also take these marrows and do 5-color flow cytometry to detect minimal residual disease. So ultimately, we'll have this comparison between genomics and 5-color flow. And ultimately, we will hopefully do stem cell transplants on patients with persistent disease evidence from one or both of these assays. So more to come on that as we proceed into the clinic with what we've learned about AML from large-scale genomics. I just want to finish by showing some actual reports from patients, just a couple here, where this is really what part, not all, of our clinical lab report looks like. And this is, of course, the diagnostic marrow for this patient. This patient cleared very, very well their tumor from what we can see in the comparison, and as opposed to this patient, which is, again, more typical, where we still have some recurrent detectable mutations even after an initial round of chemotherapy. So this is where we're headed with these AML patients in our translation of what we've learned into clinical reality, and more on that soon. I want to turn my attention now to the last part of really using the genome in a completely different way than what we've been talking about, and that is to really now enter the space of immunotherapy. For those of you who dwell in the cancer research area, you know that there's a huge amount of excitement nowadays around checkpoint blockade therapies, which are achieving dramatic results, including for our former president, Jimmy Carter, in the solid tissue malignancies. This is now just a little different twist where we're evaluating the genome for a variety of ways to identify non-self or neoantigenic peptides. So what are the underpinnings of this idea? So it's long been suggested, as you can see from the first 2 bullet points, one longer, the one at the top, than the more recent one, which was pre-next-gen sequencing, that because tumors carry unique mutations in the DNA, which ultimately turn into unique amino acids in the RNA, in the proteins that are expressed from DNA, rather, that tumors can actually look largely non-self to the immune system under the right circumstances. Now, we know from the work of people like my colleague Bob Schreiber that there are ways that there's sort of immunosuppression that prevents prevents the immune system from keeping cancers from growing out. So that's a different problem. But the bottom line is that we've known for some time that if we could identify the most antigenic non-self peptides that are produced and hopefully presented by these cancer cells to the immune system, that we might find ways of helping the body defeat cancer on its own with a little help from us. The problem heretofore has been that there weren't great mechanisms to identify what these neoantigenic peptides were, especially pre-next-gen sequencing. So Bob and I, as well as others, have now been working on this problem in the next-gen era, and I'll tell you sort of where our results are at this point in time. But really, it comes down to the ability to sequence tumors, identify mutations, and then look at the interplay between the patient's immunotype, their HLA types, and predict which of those unique peptides are actually going to bind most avidly to the immune system and elicit an immune response. So the paradigm that we use here, which I'll show you for mouse patients followed by human patients, is that if we're going to design vaccines, or indeed even predict neoantigen load as a precursor to prescribing checkpoint blockade therapy, we utilize the power of massively parallel sequencing and analysis to identify cancer-unique peptides. We identify the HLA haplotypes of the individual patient. This can also come from next-gen sequencing or from a clinical assay, either one. And we also very, very much depend on RNA sequencing data. I'll illustrate this, especially being relevant for high mutation load tumors like the methylchoanthirine sarcomas that we studied in mice, and then Also pertinent, the UV-induced melanomas that we've looked at in human patients. So the input data that we're considering here then goes into an algorithmic predictor that models the binding of these unique peptides, compares them to the wild-type peptide in the context of that patient's HLA type, and calculates the differential binding energies. So what's a better binder in the mutation spectrum than in the wild-type spectrum? And this gives us a list of tumor-specific mutated antigens, or less of a mouthful to say, neoantigens. And this can be used to design a vaccine, as I'll illustrate, or even just to predict patients, as I said, who are more likely to respond to checkpoint blockade therapy. So we developed this first in a mouse setting. As I mentioned, mice that are treated with methylcalanthine, which is a chemical carcinogen, so they have very high mutation loads, like certain human tumors such as melanomas, smoking-associated lung cancer, et cetera. And we developed this pipeline that's sort of shown here. I just described it, so I won't go into it in detail. And indeed, this was published right at the end of 2014 in Nature, where we synthesized peptides according to all of our upfront bioinformatics and then evaluated them using a variety of in vitro or ex vivo analyses, as well as looking at them in our mice patients. And I'll just cut to the chase here and show that when we synthesized long peptides that corresponded to these two mutant neoantigens, LAMA4 and ALGA8, and used those long peptides to vaccinate mice that were already growing out these methylcalanthine-induced tumors, we were essentially able to cure the mice of their tumors using a therapeutic vaccine-based approach. Fast forward to human cancers. We've studied this now in melanoma in a paper that came out in Science early last year, where we, again, took advantage of the fact that we have multiple metastatic lesions in these patients. We run these through the same sort of a pipeline, and because we're going to be going back into the patient with a vaccine to evaluate whether we can elicit T-cell responses, we also used a variety of follow-on in vitro assays rather than just trusting our bioinformatics pipeline to be right, to identify or refine our list of immunoepitopes. And then we used GMP peptides in a patient-specific dendritic cell vaccine based approach, which is not to say this is the only vaccine approach, so I'll get to that later. But in this particular case, the collaborators had a huge amount of experience using dendritic cells. So here's just a quick diagram of how we did this. You can see the sort of patient represented here. We can isolate dendritic cells from the patient and culture those ex vivo, mature them, and then mix them with these specific peptides from a GMP synthesis and infuse that back in as the dendritic cell vaccine. So essentially, the dendritic cells are carrying the specific peptides into the patient and meant to elicit a specific immune response. All of these patients, all 5 of them, had different tumor-specific mutant antigens. So this is a highly personalized approach. There's no sort of shared antigenic approach other than the fact that we included 2 control peptides in each of these patients' vaccine for shared antigens to GP100, which is frequently mutated in melanomas. So again, just a nod to heterogeneity or clonality in the disease and the importance of using RNAseq in these high mutation load tumors. You can see the number of missense mutations that were identified in these 2 patients where we had multiple biopsies. It's quite large. And when we predict binding to HLA-A*02:01, which is the prevalent HLA type in Caucasians most susceptible to melanoma, you can see that there's already a diminution in the number of neoantigen candidates. But now look at the diminution when we actually go to see which of those candidates are expressed at the level of RNA. So this is a really important culling mechanism because we don't want to waste time and money, especially on GMP peptides that are not even, have no chance of being expressed. Ultimately, would we like to do proteomics to detect whether the mutant antigens are expressed? Absolutely, and we're developing those methods now, but at this point in time, this was the best surrogate that we had. So what's the upshot? Did this work or did it not work? Now keep in mind this is a first-in-human study, so no clinical benefit was the point. Really just 2 questions. Did the patients do okay? Were there adverse events? And 2, did this actually work to elicit T-cell memory? And the answer to that second question is kinda. So if you look at the 3 patients, you can see that in the post-vaccine setting, the second column for each of the patients, in every case, 3 of the 7 tumor-specific mutant antigens that were used in the vaccine elicited a clear and discernible T-cell response in that patient by these dextramer-based assays. So we're not as good as we think we should be. Obviously, we'd like for it to be more like 7 for 7, but I'll tell you how we're going to make that better in just a moment. The answer to the other question, just to make sure that you know it or hear it, is that we did not record severe adverse events for these patients. And all of these patients had already seen a checkpoint blockade therapy, which was ipilimumab at the time, prior to the vaccination. The last thing that we looked at in this study is sort of what happened to the TCR beta clonotype. So you can see an example here in panel B for the 3 patients, just selecting a specific mutant antigen, where the dark bars correspond to the pre-vaccine TCR beta, and then the clear bars correspond to the post-vaccine expansion in the clonotype. So this is getting us a very diverse response for these patients, which is quite exciting because it means that we'll have a good, hopefully a durable response for these patients as well. So the conclusions from this study is that, we achieved our endpoints. We were able to get a partial response, and we were able to demonstrate safety. And I'll tell you where we're going from this study in just a moment. We really, I think, proved to ourselves, and hopefully to you as well, that verifying expression levels and allele-specific expression is critically important, especially in these heavily mutated tumor exomes. And we expanded the T-cell repertoire in these patients by virtue of vaccination. I don't have time to go into the details today because there are many, but we are working on ways to try and improve the accuracy of neoantigen predictions and also to refine these evaluations using blood-based evaluation of the patients both before and after they receive their vaccine. So what are our next steps in this immunotherapy genomics-guided approach? Well, there are several. So first of all, we just received, thank you, a positive funding decision from the NCI to do a second trial where we'll be combining anti-PD-1, which is now approved in melanoma patients, with a dendritic cell vaccine. And so this is really meant to sort of wake up the immune system and then hopefully elicit a durable response in patients by virtue of vaccination. So that just got approved for funding, and we'll get underway on that soon. Already open and funded by Komen is a triple-negative breast cancer trial. This is a very interesting trial design for a couple of reasons. I'll bore you with the details here for just a second. One is that we're trying 2 completely different vaccine platforms. So I already talked about long peptide cocktails, so I won't go into more details on that. The second vaccine platform will literally be piecing in the neoantigen coding regions behind a strong human promoter and then electroporating these constructs into cells in the patients and reintroducing them into the patients. So these are 2 different vaccine platforms that are being tested. The reason for triple-negative breast cancer may be obvious to many of you, but I'll just say it for purposes of illustrating that this is a window of opportunity trial. So there's one approved therapeutic regimen essentially for TNBC patients. That's ACT therapy followed by radiation after they're surgically had their tumors removed. Our window of opportunity trial then places the vaccine modality after they've had their radiation treatment, and once their immune functionality has recovered, they'll receive their vaccine. And because most of these women will recur in about an 18-month to 2-year time window, we'll essentially follow them and compare them to the norm, which is well established for recurrent disease, just to see how they do in their recurrence spectrum. So I didn't mention it, but it's essentially 15 patients per arm for a total of 30, and we're already accruing patients for this study, and indeed have sequenced, I think, on the order of 6 or 7 patients already for vaccine design. Lastly, I'll just finish with some work that we're doing in pediatric and adult gliomas that was really sort of encouraged by this study from Joe Costello's lab reported in Nature Genetics, where it was shown that in pediatric patients that received temozolomide and alkylating chemotherapy, they actually spiked a significantly increased mutation rate over their primary disease, which we know from sequencing lots of pediatric gliomas is a fairly quiet mutational landscape. So in fact, the standard of care here might actually put these patients as better candidates for an immune therapy intervention. And we're studying this now in some samples that we have banked in the pediatric brain cancer setting. I'll just show one for purposes of time, a patient that we've studied from her initial diagnosis through 3 different recurrences, all of which have been removed and banked. And you can see from the sequencing data in these different colored blocks the sort of emergence of new subclones in this 3rd recurrent tumor that essentially goes away in the 4th occurrence of the tumor and is replaced by another subclone, but definitely a role of increasing complexity over time, and obviously a case that really no therapeutic intervention from radiation to radiation and temozolomide to lapatinib Avastin, and currently the patient is being treated monthly with Avastin, has truly erased the tumor. As you can see from this small bullet point, the patient currently is well, but with a small but stable mass that could progress at any time. This patient is now 25 years old and is a very good candidate for a precision vaccine. So you can see here now the clonality ebb and flow that I just described for you with the graph. When we go and evaluate RNAseq from this patient and neoantigen potential, what we find is that this founder clone in particular, but also cluster 2 that arose in the 2nd surgery and has never really gone away either, both present candidate neoantigens for this patient's vaccine design. So I won't bore you with these details. You can read through them and ask questions about them if you'd like. There are lots of things we don't know yet about precision vaccines. So I just want to be very clear about that and not oversell this, hopefully. It's very exciting, but we just don't know things like, the patient monitoring. What's the turnaround time that we can achieve? Is it clinically relevant for patients? What are the right combinations to use with vaccines? And what's the ideal timing for immunotherapy? Do we want to use these when patients have no evidence of disease, a low tumor burden, probably not a high tumor burden, but we just, there are lots of things that we just don't know, in addition to doing a better job of predicting neoantigens. So I'll just finish by saying that I think the clinical translation of cancer genomics is really underway. I'm giving you examples from our site, but there are plenty of other places that are working hard on this and trying to make a difference in the outcomes for cancer patients. And we're doing this in both solid and liquid tumors, as I've illustrated from my remarks. And now I'll finish with the slide that acknowledges people who actually work instead of travel all the time like I do, and give talks like this. But it really takes a village. So a lot of folks in the genomic realm, a lot of folks in the cancer care and of patients, basic sciences of immunology as well, and a lot of funding from the NIH as well as private foundations. But especially we couldn't do any of this work if it weren't for patients and their families. Thanks for your attention.
Thanks, Elaine. We have time for questions. We'll start there and go back and forth.
Okay. First off, this has been very important in terms of tumor of unknown origin for those of us who have been in pathology and identification of tumors, and certainly a space-age leap over the very primitive immunoperoxidase that we used to use back in the early 80s. My question refers to your first case in terms of the hematopathological case. You talked about a bone marrow transplantation. And even if you have a bone marrow transplant from a monozygotic twin, you're bound to have a risk of graft versus host disease. And then you'll have graft versus tumor disease as well. How does that figure into the genomics, or didn't you figure that in as a specific category, or wasn't graft versus tumor a concern?
I think it was less of a concern at this point in time. It certainly is a complication that arises. There are some good therapeutics to suppress it, but my experience, and I may not be experienced enough, is that actually it's in the unrelated donor stem cell transplant where you really tend to run into significant complications in graft versus host. but it could go to individual patients as well. But we haven't really looked carefully at that from a genomics perspective.
Thank you.
Please.
Very nice talk, Dr. Mardis.
Thanks.
You know, in talking about neoantigens, you spoke about some degree of functional determination of the relevance of the neoantigen. I'd be interested in your perspective on the functional assays for point substitutions in tumors, you know, in genes like EZH2 or whatever it might be, whether there are functional assays as more people start to sequence, as whole exome sequencing gets more affordable.
Yeah.
What do you think should be done in terms of validating the relevance of these?
So that's a great question, and I think it's a question that's been spurred on by the fact that when you look broadly, you find broadly, right? And so we have like now an overwhelming number of variants of unknown significance that await sort of functional validation. I guess my only comment there is, or comments there are two. One, I think there is a great need. You know, Francis should pay attention here, but I know he knows this already. There's a great need to really fund high throughput biology behind functional assays, and certainly that's been enabled now with CRISPR-Cas and other genome editing technologies. The beauty of the neoantigen setting is that functionality takes on a completely different notion, right? So here we're now not worried about, sort of, is the protein activated, is it inactivated, but more importantly, is the unique peptide being presented to the immune system so that we can elicit an immune response. So if you look through the lists of genes that we identify, for example, in that melanoma paper that I cited, you'll basically not see a single known driver in cancer, although that's not off the table as a possibility. But typically, these are just sort of your average Joe passenger genes, where it just tends to look a lot different to the immune system based on that substitution. And I would also point out that we're really looking at the very simplest level here. So we're looking at we're looking at amino acid substitutions, but there are obviously other types of mutations, such as frameshift mutations, that would present a significantly different peptide to the immune system. And we're just now beginning to explore whether those are just fantastic, neoantigens where you would only need one or maybe two in a vaccine to really elicit a very strong response. So again, sort of adding to the number of unanswered questions, I think that certainly exists as one. And then fusion proteins, we know from previous studies in leukemias, for example, can present very unique, strong antigens to the immune system as well. So just so much more to do.
Thank you.
Thanks.
Today you focused on protein coding variants, but have you had a chance to look at the non-coding genome and transcriptome?
Sure. So we look at both. It's a lot harder to build evidence from those. I mean, certainly there have been some beautiful studies to point out the TERT, promoter mutations, which we all sort of know and love, but not a lot following on from that. I think, in an ideal world, which I don't live in but would absolutely love to, we would have not only the capability to take, for example, ATAC or DNA hypersensitivity sequencing, standard genome sequencing, transcriptome, maybe even methylation, and sort of put it all together in a big mashup to really get an integrated look at everything that's going on in that patient's genome, as well as the RNA-based ramifications coming out of it. So part of the challenge there is not just monetary, but actually, as I was telling the trainees earlier today, the bioinformatic or computational tools to really knit together that picture are not there right now today. And I think the other thing to point out, which I often do, but nobody seems to pay attention, is that we know that there's also a germline or heritable component. So we've got to begin to consider both genomes, if you will, into the equation, which is the somatic as well as the germline contribution to the disease. And I think probably the best place to start looking at that are the most simple tumors, which would be pediatric cancers, because there we really see very few mutations, and I think we can start to build some evidence around the interplay between inherited susceptibility and somatic, drivers. So lots to do, yeah.
Hi, very nice talk, Elaine.
Thanks.
You point out the fact that, of course, many of these mutations are unique, but of course there are also hot spots. And so I think when we're focusing on immunotherapies in particular, we should pay attention to those. I mean, recently in Steve Rosenberg's group, we found that KRAS can be targeted, a specific KRAS G12D mutation, and we also are working on developing T cells that recognize KRAS G12V mutation. So many of the variants and the hot spots really, I think, serve as very good targets, not only from the fact that they're shared among multiple tumors, but also their driver mutations. As you point out, you don't want to target things that are these subclonal mutations. The driver mutations, of course, generally are clonal. So I think that's an important thing to point out, that we should be really focusing on those as well.
Yeah, with some caveats. So I may not be so well versed in this, but my understanding in terms of the KRAS mutation was that there was also a highly unique HLA haplotype for that patient that made that a neoantigen, which wouldn't be the case for everybody. I mean, I think at the end of the day, we'd all love to find the ideal shared antigen, and by golly, if that were KRAS G12 or 13, pancreatic patients would be beating the doors down, among others. I mean, those are fairly rare instances that, may pan out over time, but I think most of the look across the genome hasn't really turned up these, sort of shared antigens as yet, and may not. So, the personalized vaccine approach is a little bit of an odd duck because it's extraordinarily personalized. And, we really have to convince ourselves of answers to all of these questions, as well as a clinically relevant time frame to come up with these modalities. And that may not work. I think it's worth pursuing, but it actually may not work on a lot of levels. So, yeah.
Well, Elaine, it's wonderful to watch your transformation into an immunologist. This is exciting to see these fields coming together, and you represent it well. Those of you who might like to continue the conversation, there'll be a reception in a minute in the library. Please come and have coffee and cookies and engage Elaine for a brief time before she has to run off to the airport. But meanwhile, please, let's thank our speaker again.
Thanks very much.